Good afternoon, students. We are discussing the chapter Principles of Inheritance and Variation, isn't it? So, in our last class, we had discussed the law of dominance, which is the first law of Mendel. Hope it is clear to you all. And now, today, we'll discuss the law of segregation, or we can say Mendel's second law. So this law is also based on the results of Mendel's monohybridization experiments. So in our last class only we had discussed that the first two laws, means the law of dominance and law of segregation, these two are based on Mendel's monohybridization experiment. And the third law, which is the law of independent assortment, it is based on dihybridization experiment or dihybrid cross. So what is law of segregation or Mendel's second law? So it states that in the hybrids or heterozygotes of F1 generation, two contrasting genes or alleles, though remain together for long time, they do not contaminate or mix or blend with each other, but they separate or segregate at the time of gametogenesis, so that each gamete receives only one gene of either dominant or recessive nature. So we all know what is a monohybrid cross, and we know how to find out the F1 generation, and from F1 generation how to get the F2 generation, isn't it? So according to the law of segregation, in the hybrids or heterozygotes of F1 generation, so if we consider the stem height character, I am giving one example. So if we consider the stem height character, so here in the parental generation or P1 generation, this is the P1 generation. Last class I have shown in detail. So then we get the gametes of P1. So today I am not writing because in our last class we had already discussed. Then we'll get the F2 generation. Again this capital T small t, capital T small t, capital T small t and capital T small t. Now in law of segregation, in the hybrids or heterozygotes of F1 generation, two contrasting genes or alleles do remain together for long time. They do not contaminate or mix or blend with each other, but they separate or segregate at the time of gametogenesis. So I am explaining how. So this is the hybrid of F1 generation. So here in the hybrids of F1 generation, here two contrasting genes are present. One is capital T, means tall gene for tallness, and small t for dwarfness. So in the hybrids of F1 generation, though these two genes remain together for a long time, but they do not contaminate or mix or blend. That means they never mix up. And at the time of gametogenesis, what is gametogenesis? The process of formation of gametes. Okay, so if we self-fertilize the F1 hybrids, Okay, this is self-fertilization of F1 hybrids. What will happen? These two contrasting genes, gene for tallness and gene for dwarfness, they never mix or blend with each other. All the genes are pure, and at the time of gametogenesis, that means during the formation of gametes, they get separated so that each gamete receives only one gene of either dominant or recessive nature. কি হয় F1 হাইব্ৰিডবিলাক আমি যেতিয়া সেলফ ফাৰ্টিলাইজ কৰোঁ যদিও তাত দুটা জিন থাকে দুটা কন্ট্ৰাস্টিং জিন থাকে এটা টলনেছ এটা ডুৱাৰ্ফনেছ তথাপি হিহঁত কেতিয়াও মিক্স হৈ পেলাই এটা মিডিয়াম হাইটৰ কেৰেক্টাৰ এক্সপ্ৰেছ নহয় হিহঁত সদায় তেনেকেই থাকিব এটা কেৰেক্টাৰ এক্সপ্ৰেছ হয় এটা হিডেন হৈ থাকে আৰু গেমেট ফৰ্মেশ্যনৰ সময়ত হিহঁত সেপাৰেট হৈ যায় যাতে প্ৰত্যেকটো গেমেটে এটা পিওৰ জিন পায় সো ইয়াতে gene for tallness, gene for dwarfness, again gene for tallness and gene for dwarfness. So, the mechanism of law of segregation can be explained with the help of a monohybrid cross. It is obvious that though in F1 hybrids only the dominant phenotype appears and the recessive phenotype is not lost because it reappears in F2 generation. This results that although the alleles of dominant and recessive nature, these two, remain together in F1 hybrids for a long time, there is no mixing or blending of the characters. At the time of gamete formation, these two alleles or factors obviously separate or segregate. Otherwise, recessive characters cannot appear in F2 generation. So, we know that in F2 generation, the offsprings, I am showing the F2 generation, capital T, capital T, capital T, small t, capital T, small t, small t, small t. So, in F2 generation, what happened? One dominant plant or homozygous tall, heterozygous tall and homozygous dwarf. That means the recessive character is expressed in F2 generation. If the genes or characters mix or blend in F1 generation, then in F2 generation we cannot get the recessive character or we cannot get the recessive plant or dwarf plant, isn't it? So F1 hybrid বিলাক যদি ইহঁতৰ জিন্স বিলাক যদি মিক্স বা ব্লেন্ড হৈ গোল হয় তেতিয়াহ'লে আমি F2 জেনেৰেশ্যনত কেতিয়াও পিওৰ ডুৱাৰ্ফ প্লান্ট এটা নাপালো হয় ইহঁতৰ মিক্স নহয় কাৰণে জিন্সবিলাক গেমেট ফৰ্মেশ্যনৰ টাইমত সেপাৰেট হৈ যায় কাৰণে আমি F2 জেনেৰেশ্যনত পিওৰ ৰিসেসিভ প্লান্ট এটা পাওঁ সো এই লটো এক্সপ্লেইন কৰিবৰ কাৰণে আমি ইমানখিনি দেখুৱালে হৈ যাব till this much is sufficient to explain for the explanation of the law of segregation okay till gametogenesis this much is sufficient so here we see that gametes produced at any time always carry either the dominant or the recessive allele, either the dominant or the recessive allele. This is why it is called either as principle of segregation or law of segregation or law of purity of gametes. Okay? So law of segregation took law of purity of gametes বুলিও কোৱা হয় কিহৰ কাৰণে because gametes are always pure তাতে কেতিয়াও gametes বিলাকত mixed characters বা mixed gene নাথাকে যদিও F1 জেনেৰেশ্যনত আমি হাইব্ৰিড পাওঁ তথাপিও হিহঁতৰ মাজত কেতিয়াও মিক্স নহয় আৰু আমি পিওৰ জিনবোৰ পাব পাৰোঁ এক্সামত কেতিয়াবা কুৱেশ্যন আহে why law of segregation is also called law of purity of gametes so তেতিয়া আমি এইখিনি কথা এক্সপ্লেইন কৰি দিব লাগিব গেমেটোজেনেসিসৰ কথাখিনি ওকে now we'll discuss back cross. What is a back cross? The cross between F1 hybrids and either of the two parents is known as back cross. So we know F1 hybrid for the stem height character is capital T and small t and their parents capital T and small t. So this is parental generation, this is F1 hybrid. Now back cross namtur pore ami gom pao it is we have to look back. So here this is F1 hybrid. So when F1 hybrids are crossed with either of the parents jikono eta parentor logot jetia ami F1 hybrid or cross koru take back cross buli koa hoy so moi dekhuaisu now I am crossing the F1 hybrid with the dominant parent so when F1 hybrids are crossed with the dominant parent all the F2 individuals express dominant character. So I am showing how F1 individual by F1 hybrid or logot jeti ami dominant parent or cross koru F2 individual ot ki hoy gute keta dominant character e express hoy. So ibla hoise gametes capital T again capital T capital T small t capital T small t so here look two homozygous tall and two heterozygous tall plants but their phenotype though their genotypes are different one is homozygous two are homozygous and two are heterozygous but their phenotype is same means all plants are tall so when F1 hybrids are crossed with the dominant parent all the F2 individuals will express the dominant character okay now when F1 hybrid is crossed with the recessive parent then what will happen moi duitai dekhwai disu jete recessive parentor logot cross korim ami F1 hybrid because duitai back cross hoy so here so capital T small t capital T small t small t small t small t small t so in F2 generation both the parental characters will appear in equal proportion so yate tall duta tall plant aru duta dwarf plant produce hobo. So 2 tall plants and 2 dwarf plants will be produced. So both the character or both the parental characters will appear in equal proportion. So this is back cross. Is it clear? So here genotypic and phenotypic ratio is 1:1. Genotypic and phenotypic ratio and ratio 1:1, because here also in this case genotype 2 heterozygous capital D small t and 2 homozygous dwarf plants. So 2 tall plants and 2 dwarf plants, phenotypic ratio is 1:1. 1 heterozygous and 2 heterozygous and 2 homozygous, that is genotype, again the ratio is 1:1. So here both the genotypic and phenotypic ratio 1:1. That means the ratio is same. Is it clear? So go through it. Thank you.